Good evening, my name is Jeff Levier. Today we're going to talk a little bit about post-mortem staining occurring within decedents. One of the things I want to start off with is comparing livor mortis with post-mortem staining. Because a lot of times when you talk to people outside of the industry, they just kind of assume that it's the same thing. And it's really not. It's two totally different things. So we're going to kind of just run through some of the differences real quick. Livor mortis is considered intravascular, which means it's occurring inside the blood vessels, whereas postmortem staining is extravascular or occurring outside of the vessels. And rigor mortis can occur as early as 20 minutes after death, whereas postmortem staining normally takes 6 to 12 hours before you'll notice it after death. Livor mortis is known as a physical change because we're just relocating the blood and there's no change in the composition of it. Whereas postmortem staining is considered a chemical change because we're actually changing the makeup of the blood. A livor mortis can be removed a lot of times by the use of the arterial fluids used in embalming, whereas postmortem staining, the embalming fluids will not remove it. It might lighten it a little bit, but it will not take it away. And then livor mortis occurs by hypostasis, which is the blood settling into the dependent parts of the body. And then postmortem stain occurs by hemolysis, which is the breakdown of red blood cells, relaxing the hemoglobin into the surrounding fluids. When pressure is applied in a livor mortis spot, the blood is going to move away from the area where the pressure is applied, and you're going to see the clear spot underneath. Whereas post-mortem staining, if you use your digit and apply some pressure, it's still going to remain, the color is not going to move away from the pressure point. One thing is important to note that post-mortem staining can occur before, during, and after arterial injection occurs. It can also occur in combination with livor mortis, even within the same tissue regions. Now, what role does heme play in postmortem staining? After death, heme, which is an iron-containing substance, is released from red blood cells as they break down, and it seeps into the surrounding tissues. A chemical reaction occurs whereby the iron oxidizes and can create a deepening discoloration. Postmortem stain creates challenges for the embalmers. First off, it creates additional challenges for an embalmer to restore the natural appearance of the decedent. It requires additional time normally to complete the embalming process. It requires the use of a stronger arterial fluid, sometimes with special bleaching agents added, as well as you may have to inject individual tissues and use of surface packs to help reduce the discoloration. Factors which affect postmortem staining, in addition to how it affects embalming, different situations require different things. Such effects that can occur can be based on cause of death, the blood chemistry of the volume blood at the time of death, length of time since the death occurred, condition of the body, environment the body was found. So all of those can create some additional challenges for embalmers, and they really need to be evaluated on an individual basis. Another interesting thing about the postmortem is the type of poisoning can be revealed through the distinct coloring of the postmortem stain. If the stain is cherry red, it can be kind of assumed, or that may be common, that it was a carbon oxide poisoning. Or if the staining is pink, cyanide poisoning. Or even brown could be indicative of a nitrate poisoning. So the colors can definitely tell us a little bit more about the scenario as well. And lastly, I included some references on this slide due to our limited time we have this evening. If you'd like to learn a little bit more, delve a little bit more deeper into the postmortem staining, then please utilize these references. And I appreciate your time and hope that you found this a little bit enlightening for your learning experience.